Yeah, I mean, my question to Carl was not entirely innocent because what I've been talking about is a functional anatomy of language, which is already a very, very large concept because it actually covers many different functions. I wanted to start with a small historical perspective. This is the functional anatomy of language viewed by the neurophonologist years ago with language localized somewhere behind the eyes, and it was very symmetrical. So, your analysis should show up something very symmetrical in these areas. But then it took about the same time what we name the neuropsychological approach, which is connecting lesions with brain functions to actually show that there were some relationship between anatomy and language. This is the first case described by Paul Broca in 1861 of a patient with a loss of articulation, so he couldn't pronounce but one single phoneme was 10, and that particular big lesion of the inferior prefrontal cortex, which actually extend towards the white matter inside. So it's a much larger lesion than just the pure frontal lesion, but still it's the first case that describes a strong connection between anatomy and language function. Now, we've been progressing into how language actually implements in the brain through two different ways in patients. First was the work by OHBM. We did electrical cortical stimulation of awake patients, and stimulating until he observed a stop in the function. So basically, here you're in open school surgery, and you introduce electrode and you stimulate the cortex different places. And every time you find a place where you stop the function, you say, this is the site of language. Okay. The other approach is mapping the lesions in the stereotactic space that corresponds to aphasia. Would that be, production aphasia or comprehension aphasia? And by doing this probabilistic mapping of lesion, you have one aspect of how language is implemented in the anatomy. Interestingly, this paper by Turken and Dronkers years ago showed that by mapping this language area through lesions, you can actually recover some of the language network. And for my talk, we really focus on this network aspect, because in the previous talk, I thought we really focused on specific areas in the cortex that were connected regions. I believe language, or even very higher cognitive function, implements through network activity. And that's where also Carl's talk is so important, because it shows that although you cannot really connect one function with one receptor, or one function with one layer, or one function with one type cell, what really is important is the network aspect. And some of these higher connection functions express themselves through network activity. This is the first example of language and functional neuroanatomy in the literature done in human subjects awake in healthy using PET. This is a study by Steve Peterson in 1988. It's a study of cortical anatomy of single word processing. So here the study was just to ask people to just see a word or hear a word, pronounce a word. And so the idea of this study is trying to map onto the cortex which are the part of the cortex involved in this very simple, so-called simple task of just hearing a word and understanding what that word means. This study shows clearly that there were some consistent data between this neuropsychological data and the functional imaging data, and showing activity for the semantic part of this language task, which is understanding what the word means, in the inferior prefrontal cortex. So that was more or less matching the Broca case and also the Dronkers lesion mapping. One thing that was missing in this first study was an analysis of how asymmetric is this system. That was the sense of my question to Carl. One particular feature of language in the brain, at least in healthy subjects, is that most people in this room have language implemented in the left hemisphere, meaning that it's mostly regions of your left hemisphere that are involved in producing and understanding language. So we've done that study years ago where we actually mapped different language components from phonology up to higher semantics and show that asymmetry was a characteristic of the language in the brain, and it varies with language component, but also varies with language area. So language is not associated just with one brain region, and asymmetry is not implemented in one particular brain region. It really depends on which region is involved in which language component. We started that recently with a big database. I will come back later with 300 subjects. And if you use this big number of subjects with a lot of left-handers, so you can actually not bias your sample by using only right-handers, and you just map language production, so it's a contrast between hearing a sentence in your head or hearing just a list of words, and using this simple hemispheric functional laterality index, which is how much activity you have in the left hemisphere, how much activity you have in the right, and make the difference. So positive here means that you have more activity in the left than in the right. You see that most right-handers have higher activity in the left, but it's also true for left-handers. So really the hand doesn't really connect with the hemisphere you use for language. So that's one point. Second point is, if you really try to understand the relationship between anatomy and function, then you can also go to meta-analysis. So it's been recently and multiple times recalled that the human brain is highly variable. Svenja showed that you have high variability in the gyrification. You have high variability in cell density. You have high variability in receptor density, but you have also high variability in the way each function is implemented in each individual. And if you go across labs and if you go across studies, then people have been trying to do meta-analysis of language localization. So this is a study done in our lab years ago with 164 studies from the literature. You take the peak of activation, you map them on the left hemisphere, and each of these dots is one study. Okay, and so you can see that there is a rough distribution of the study across the inferior frontal. You have also the motor cortex, and then the temporal cortex, and part of the carrefour, the occipito-parietal junctions. But this is highly variable between studies because you combine variability between individuals, variability between machines, labs, protocols, and so forth. Still, there is sort of consistency of the language system, although you have many problems with this meta-analysis. First problem is because you have all these different language tasks that are chosen by the different labs, you have a lot of dispersion. You have a mixture also of low and high contrast in the contrast map, and you have variability in samples and imaging protocols. The other thing is, everything I've shown you so far is just left hemisphere. So when people analyze language distribution and anatomy, they always look at the left hemisphere activation. Nobody's really looking at the symmetry. Also, we know that if you look, for example, at the just a pure metabolic map at rest, you will see that the right hemisphere is reactive at rest. So is the left hemisphere. And it's only when the function expresses itself that there is this imbalance. So asymmetry gives you information about the language areas. And finally, which I think is important, is we don't have really information about networks. So what you've seen here, when you do this meta-analysis, or when you do even the activation study, what you have is a map of the activation. You don't really look at the network activity. So the question we ask ourselves is whether we were able to look at language networks, mapping the different regions that work together to produce language. So for this, we developed a method that combined in the same people using different tasks of different language tasks, and in that, all these tasks assess the asymmetry as well as the activation. And second, combine this with resting state activity. Why did we do that? First of all, I'm sure you're aware of this work that claim that when you look at resting state networks, you actually look at a sort of surrogate of task-related networks. The idea being that activity at rest is a reflect of what will happen when we will actually do functions. It's not entirely true. If you look at the resting state network, they don't really match cognitive networks. But there is some overlap between these resting state networks. I'm not talking about the DMN. I'm talking about all resting state network and the cognitive networks. So a paper by Steve Smith, for example, that shows that. So one idea would be that if you have the language networks, language regions, maps using activation, then you could extract some dynamic information from the resting state network associated with resting state activity associated with these language areas. Then, to identify the sentence-specific regions, forgot to tell you that here we will focus really on the sentence comprehension network. So to identify one specific component, we will use first a very large sample of right-handed participants. Then we will contrast this task. And for this, we use this database that we'll be building. It's a very specific database. It's a database done to study lateralization, so 16 different tasks done in 144 subjects, all the same. And is there a systematic way of producing the next slide? Yeah. And then we will use specific methods to look at the connectivity between these regions. So you can look at connectivity with high resolution DTI, but here we will look at functional connectivity. How we define this, I will spend just a few slides to explain what is this new atlas we've been using, Aisha. It's an atlas that is based on resting state activity and on the observation that it's at rest if you look at the correlation between activity, bold activity, metabolic activity between brain regions. The most correlated areas are homotopic areas, with activity going back and forth between regions through the corpus callosum. And finally, the last step will be to do conjunction analysis of all this data, okay, because we want to cancel out the activity due to the lower control. Okay, let's go. So the Billie Jean database, that database that we published 2 years ago, it's a database about hundreds of people with a cognitive battery and a big functional battery. And for these 300 subjects, we have this language task battery done in all subjects. Nothing. So this is focusing just on the language that we've been using. Here we have a run of production, a run for listening, and a run for reading. So we have 3 different modalities for language. And in all cases, we've been using a visual stimulus, which is a little cartoon. And on that cartoon, the subject has either to do a covert production of a sentence, or to read a sentence that appears on the screen, or to listen to a sentence that corresponds to the cartoon. And the control task is always the same, which is on a scrambled version of the cartoon. It will just recite and overlearn a list of words, or listen to the list of words, or see the list of words. So this way, we always contrast things that are at the same level for the task or for the control task, and on the same subject for the 3 events. So we control as much as we can the variability between task and subject. And for each of these tasks, we will derive the asymmetry index and the bold variations between pair of homotopic regions of interest. So now let me explain. This is our view of how we do neuroanatomy for higher language cognitive functions. So here, the idea, if you go whatever atlas, you have asymmetric region of interest. If you look at your atlas, if I use our AAL atlas, which is an atlas based on gyrification, you have asymmetry, and it's very hard to correct for this, either at the volume level or at the surface level. So we know that we want to use an atlas of regions where we are sure that we're looking at regions of interest that are functionally identical. So for this, we use the resting state data. So in the same 144 subjects, we use the resting state data, we use independent component analysis, and we extract a number of what we name resting state networks. Okay, these are the networks visualized here. And now what we do is for each voxel, we do a profile of these different, how this voxel project, is time course project on these different networks. And for doing so, and doing clusterization, we are able to actually cluster voxel with similar activity pattern during resting state. Okay. And now what you can do in the next step is look at, in each hemisphere, which cluster maximizes the correlation with the cluster in the other hemisphere. And if you do that, then you end up with, then you can define the optimal number of clusters. This is some criteria, objective criteria. You end up with a parcellation of your entire brain in homotopic, functionally homotopic areas. So this is the atlas that Marc Joliot has recently defined, and it looks like this on the surface. It also has regions homotopic at the gray nuclei level, and it's a sort of refined parcellation of the aerial atlas. So you have much more regions, but each of regions are by pairs. Okay, one in the left, one in the right hemisphere. And if you look at how it compares to other atlas, so here what you have is an index that describes the regional heterogeneity of the pairs of region, meaning how do the left and right region looks to each other. And so obviously, like the Craddock atlas, the regions, the atlas that are based on functional correlations are better and more homogeneous than any other atlas that is based on anatomy, just because you constrain your regions to have a similar pattern at the functional level, not at the anatomical level. Okay, how do we use Aisha now? So we will measure the activation in these pairs of regions, and we will also measure the asymmetry in these pairs. So we can define the left minus right asymmetry in each of these pairs of regions. And then we will do conjunction analysis. We will extract in the 3 different contrasts for production, reading, and sentence the asymmetry. So this is what comes out at the first level of analysis. This is a set of Aisha pairs of regions where you have activity on the left in all 3 contrasts, okay, and also leftward asymmetry, which is in this language task, activity is significantly higher in the left and significantly activated in the left. So at the first gross level, you find again this big networks looking at the temporal cortex and the frontal cortex, but you have also other activity within the gray matter, the gray nuclei, and also at the parahippocapal and hippocapal regions. So that was the first step. The second step is now, can we extract, this is still a view of language areas, can we extract networks out of this? So to do this, we use this resting state activity. We will, first of all, extract for each participant the profile of resting state activity in these areas. So we use all voxels, and for each subject, we average the resting state activity in this. Recall that each of these regions are Aisha regions, so they already have an internal consistency in terms of connectivity with the other hemisphere. And then we will build the intrinsic connectivity matrix of these 32 areas, and we will just apply agglomerative hierarchical cluster analysis on this resting state activity in this activity matrix, try to extract from these 32 groups of regions with similar time pattern during resting state. So the ICA from this 32 by 32 matrix extract 4 main networks. And these 4 main networks are like this. First, what we name the core, line core, which is a core network for language areas, has, I'll show you after that on the surface display, as most important regions in the frontal and temporal network. Then we have what we name the line default mode network, which are, there are a set of regions that come out from this. Remember, these are regions that are activated and asymmetric during language. But still, these regions, they belong to the DMN, what Carl has been talking about a little bit before. And then we have 2 other networks, which we name line visual and line executive, which are networks that are part of the task, because each of these visual tasks and language tasks has some common denominators. For example, in each of these contrasts, you have to switch between the activity of, for example, producing the sentence to the activity to producing the list of words. Or when you hear a sentence, so there is some switching activity that requires some executive function. And also, although we have this high contrast, processing the cartoon requires some more visual activity than just processing the scrambled version. Just to show that you can actually, from this set of regions that are defined at the functional level, cognitive functional level, you can extract significant networks if you based your functional analysis of connectivity on the resting state activity. So there is something really profoundly true in the fact that the resting state networks or the resting state activity contains the connectivity or part of the connectivity that will be used for producing the function. Maybe I can pass this. I don't know. I have time. So we evaluate this procedure in 2 different ways. The first thing we did is using multi-scale bootstrap presampling. So we stimulate the data set and we show that each network is highly reliable. So if you stimulate this data set, you will find these networks again and again. The other thing is how much it depends on the clustering methods. We've been doing 4 different clustering methods, and most of the time we just find again the same clusters of region, especially the long core, which we name the core network for language. So I think it's pretty robust in terms of findings. Then this is a display of the 4 different networks that we found. As I told you, this first network, this anterior-posterior network, is really the core network for semantics. So I point this because if you remind the figures or the illustration that came out from this meta-analysis about covering almost the entire left hemisphere, you can actually select very precisely using this approach. The different parts, the different networks that are playing within your task. Second thing here is that these different networks are also characterized by different components, obviously, and you can sort of precisely match what they are used for. As I told you, a line core is really the semantics part of it, and it will need more work to just define exactly how is this structured. I'm not sure that you can just have the complete picture just looking at fMRI data and resisted data, and that we might need more and more refined protocols to actually understand what are the different role of these different components as a network, how semantic function actually emerges from these different areas. But at least you can start cleaning this entire picture by removing components that then you can assign to other process occurring during your task. The second thing you can do is quantitative analysis. Here is an analysis of how the different networks, which is core, visual, DMN, and executive, progress in terms of both signal variation, both in activation and asymmetry. And one thing that is clear from this picture is that it's the core network of language, this network of frontal and temporal areas, that actually capture most of the activation and the asymmetry. Meaning that, again, it's not just by looking at what pops up, but the intensity of what pops up, whatever that means in terms of neural activity, and the asymmetry, because line core is by and large the most asymmetric area that really helps you localizing, the networks that are involved in your task. Then, in the last step, what we did is trying to characterize the relationship between these different components. So the first thing we did is we've been looking at the correlation between the intrinsic activity, the resting state activity of the different networks. So here, you basically average resting state time course for each voxel of each network. And so that gives you a time course for the network. And then you look at the correlation. And this is just summarized on this slide. So what you see here, the core network of language, let's say the semantic network, is very highly correlated with the executive network, which is to produce that semantic function. You need also the executive network, but it's negatively correlated with the DMN part of it, and almost very small correlation with the visual network used for producing that function, which makes sense. It means basically that this function, this semantic function, is roughly independent of the modality you'll be using to produce it. And then finally, we did this graph analysis, because once you have your network, you can actually look at, for the core network, look at how important are these different regions in terms of exchanging within the network. And here came out a very interesting result. So we used two different indexes of graph analysis. One is degree centrality. So degree centrality measured the local activity of one particular region with respect to the other region of the network. And between centrality, look at how important is one region with exchange outside the network. And so what comes out in this picture is that three regions are very different from the other regions, the other 13 regions of this core network for language. The three regions are localized, one in the triangular part of the inferior frontal gyrus and on the posterior part of the superior temporal sulcus. So here we have what we name hubs in terms of network, and they're very different from the other region. And what it means is that you have the possibility here to identify, we believe, what the neuropsychologists or neurologists name essential language area. Essential meaning that if you have a lesion of this in this area, then you will have a dysfunction or a loss in the function of semantics. And so all over, I think the other final explanation that I want to give you is that this degree centrality explains the activation amplitude. So that's another way of looking at this data, is just by looking at the resting state activity in your areas, you are able to predict how big will be the amplitude. And that's a question for you, Carl, also, is this is going back to physiology. We have here degree centrality. We call it's the importance, the number of connections that one region has with other regions of that network. And that predicts the importance of the activation in that region with respect to the other one. So there might be some mechanism, by which we could explain this. I don't know if it's at receptor level, if it's at just the connectivity level, but this is a result that I think is very important, demonstrating that in a way you can use resting state data, map on the language areas to actually study how big will be the importance of the activation. So, in conclusion, we've been able to identify language networks rather than language area, okay, using a combination of task-related activation and resting state activity. The other point is we found that there is a core language for semantics. I mean, I should say semantics because the contrast really mapped out semantics. And that we found that the line network, the core network, was very strongly correlated with this executive network. We also found that there were hubs within this core network of language, and that these hubs were actually mapping what we would name Broca and Wernicke area, which I don't like, because Wernicke area, if you look at literature, it's just impossible to define, as is Broca area. But at least we can localize them at the level of the triangularis part of the inferior frontal gyrus and in the posterior part of the superior temporal sulcus. And these are hubs. Okay, so they are highly connected within and between the outer regions. It would be very, very interesting now to map other kind of data, including high-resolution DTI, cytoarchitectonic, and receptor data to refine how this language core network is working, apart from the fact that we have 3 hubs and other regions. And finally, we think that what we provide here is sort of atlas of core language area that can be used when you cannot do activation study. Let's say, for example, you do developmental studies in patients where it's very hard to do actually functional studies because you're sort of biased by the way the patient or the kid will do the task or maybe unable to do the task. You can actually map resting state activity on language area, but you could do the same work for other higher cognitive function and actually study connectivity and see how we can predict that connectivity will end up producing activation if the subject was doing the task. And so we think also that one outcome of this work is that we have new phenotypes to describe functional activity that goes, without functional protocol by themselves. So I think we can also work on this in the future. Just wanted to mention that this is obviously a work that's piloted by Nathalie, who didn't come, and just wanted to mention this guy, Loïc Labache, who is a PhD student working all this graph analysis, and Marc Joliot, who is doing the resting state data. Thank you very much for your attention.
Yeah, thank you very much. We have time for